So these are a series of revision slides for Biology, Unit 1 Applied. So I'm going to talk my way through and explain what I'm expecting from you. So first off is all you have to do is label these plant and animal cells and kind of discuss whether they're in a plant or an animal. Okay, pause the slide, have a go, and then in a minute the answers will appear. So you can see on here, and I'll zoom in for you and you can kind of pause because I don't want to spend ages talking through things that you can see. Chloroplasts, cell wall, vacuoles, things like that. Okay, so nucleus, just a couple of things, contains the DNA. Okay, cell membrane controls what exits and enters. Okay, cytoplasm is where chemical reactions take place. Vacuole contains the cell sap. Chloroplast is the site of photosynthesis. And then cell wall is for making a plant cell rigid. Okay, so what hasn't been included in these diagrams is mitochondria, which is where we respire, make energy. Okay, so by here then is a little bit of these 4 in all of them, these 3 we'll only find in a plant cell. Okay, and you will need to know the differences and things that go on in them as well. So exactly like what we've just discussed now, functions of all the different parts of the cells, and then a little bit on osmosis and diffusion. There's a little bit down here on specialized cells and a little bit on cells and tissues and organs. So again, pause the slide and have a go. So I've kind of talked through this on a previous slide, but I will again. Cell membrane controls the movements of substances in and out of the cell. Cytoplasm is where chemical reactions occur. Nucleus contains the DNA. Mitochondria, where respiration takes place, releasing energy for the cell. Vacuole then has the cell sap and keeps the cell firm. Cell wall, tough substances called cellulose supporting the cell wall. And then chloroplasts contain the green pigment chlorophyll, which absorb light energy. Okay. Osmosis then is the diffusion of water, so the movement of water from a high concentration to a low concentration through a partially permeable membrane. So a membrane that's big enough to fit small things through, but not big enough to fit big things through. Okay. Diffusion then is when particles spread, again, moving from an area of high concentration to low concentration, particles are free to move, there's no membrane. Specialised cells then, cells that are differentiated to carry out a particular function. I've just included the red blood cell because they have a biconcave shape and no nucleus to fit in loads of oxygen. And I did sperm cells because they've got a tail that can swim. Cells, tissues, organs. So cells are the smallest bit of your body. So we're made of animal cells. They all group together themselves to make tissues. Them tissues group together to make organs. Them organs group together to make organ systems, like the circulatory system, respiration system, digestive system, all these types of systems. And then all together these systems work to make us as an organism, a human. Respiration then, there's a bit of a labelling of a diagram and then some questions to answer. Again, pause it and in a minute I'll go through the answers. Okay, so here's a label diagram of your respiratory system. You could be asked to label any part of this. Be careful with things like intercostal muscles, ribs, okay? It goes trachea, bronchioles, bronchus, alveoli, okay? You need to be able to talk about the alveoli, diaphragm, ribs, okay? What is the need for respiration? For muscles contraction, that requires energy for growth and repair. So anything that requires energy in our bodies is what we would be using this for, okay? Equation for aerobic respiration, so glucose, that comes from our food, plus oxygen, which comes from breathing in, then carbon dioxide is a waste product, water and energy is the big thing that we make, okay? Anaerobic respiration then, glucose, no oxygen, makes lactic acid, which is what causes cramp, and energy in the form of ATP, which results in oxygen debt. That's anaerobic because there's no oxygen. The cramp, you need to take really heavy breaths then to get rid of the oxygen debt. Which respiration makes more energy? Aerobic respiration. Breathing in and out, and looking at the alveoli again. Pause the slide, have a little go, and then I'll run through the answers. Okay, so what happens when we breathe in? What happens when we breathe out? Okay, so when we're taking a deep breath in, you'll be able to feel this. Your ribs move up and out. Okay, your diaphragm flattens, your intercostal muscles contract, which allows the chest to increase. Okay, that means that the volume inside the lungs is increased, lower pressure, so the air rushes in. When you breathe out, then your ribs go down and in, your diaphragm moves up. The volume of your chest decreases, so decreased volume, high pressure forces the air out. Alveoli then. So alveoli is right at the end of your lungs, which is where gas exchange takes place, okay? CO2 comes out of your blood and you breathe it out, goes all the way back up to your lungs. O2, that you've just taken a deep breath in, then goes into your bloodstream, okay? How is it adapted? It's got thin walls, just 1 cell thick, a large surface area, it's moist, and a rich blood supply. So, digestion, here's a diagram of your digestive system. Again, you have to be able to label it. And I put a series of questions on the side, and you can have a go at them. Again, the answers will appear in a minute. Okay, what is the purpose of digestion? To break down food into smaller, more useful products. Where is water reabsorbed? In the large intestine. Where is bile stored? These questions come up quite a lot. Bile is stored in the gallbladder, is made in the liver. What is the function of bile? It neutralises stomach acid, increases surface area for digestion. Where are nutrients reabsorbed? Small intestine. What happens in the stomach? Food is churned and broken down. Function of the pancreas to make digestive enzymes. How does food move through the system? Peristalsis, okay, make sure you know that word, it's just muscle contractions. Why do we have acid in our stomachs? To kill microorganisms. What is egestion? When feces pass out the body. So, food tests and enzymes then. So there's 3 enzymes you need to know, running down here, and 3 food tests that you need to know. Okay, again, pause the screen, have a go. And I'll put the answers up in a minute. Okay, so to test for starch, you need to add iodine. If starch is present, it will turn from yellow to blue-black. Okay, to test for sugars, and we tend to mean glucose, that's the big sugar that we mean. Okay, add Benedict solution and heat, that's really important. It turns from blue to brick red. Okay, lipids, or sometimes we call them fats, okay, add ethanol and it'll turn cloudy, and then proteins, add burette. So notice them words are very similar, okay, burette, and the blue solution turns purple, okay, you need to know that. Okay, protein then is broken down into amino acids using protease, so we often see this. Remember, protein is the long chain, and then amino acids, okay, are the little ones then, the smaller ones. They fit through your cell walls. So protease is the enzyme that does this, okay. Starch then is broken down into glucose using carbohydrates. There's a spelling mistake, that should be an S, not a T. And then fats and lipids are broken down, and there's 2 here, fatty acids and glycerol using lipase. So this slide is all a lot of learning, but you need to know it all. And then the final little bit for the biology section is what is an enzyme. So there's a little bit of a past paper question to have a go at here. Okay, so you can fill that in, and then there's 1, 2, 3 questions to have a go at as well. Again, pause it, have a go, and then I'll fill in the answers as we go. So the first thing here, it says complete the diagram below to show the staged reaction between the simple molecules. So you need to add them in to the active site like that. Okay, so they're all added into the active site. This is called a lock and key. Okay, so it's a lock and key. What is an enzyme? It's a biological catalyst, a substance that speeds up the rate of chemical reaction without being changed. This is a key definition that you need to learn. How can an enzyme be denatured? Change in temperature or pH. If the shape of what define denature? So it's when the shape of this changes. So if it got really hot, that wouldn't be able to fit anymore because the shape would have changed. Okay, so it will no longer work.